Hello everyone. Our today's topic for discussion is translation, which is also known as protein synthesis. The biosynthesis of a protein or a polypeptide in a living cell is referred as translation, and translation can be divided in following stages which we are going to discuss. The first is what are the requirements of the components for translation. Second is the activation of amino acid. Third is the protein synthesis proper, and then we will discuss about the post-translational modification. So, what are the requirements of protein synthesis? The first and important requirement for the protein synthesis is ribosomes. Ribosomes are the centers for the protein synthesis. They are also called as factories for the protein synthesis. Ribosomes are made up of proteins and the ribosomal RNA. Each ribosomes have two subunits. The first is the big subunit and the second is the small subunit. Ribosomes have two functional sites, the A site and the P site. The A site binds with the amino acyl tRNA and the P site is for binding peptidyl tRNAs. A site is the acceptor site and the P site is the donor in eukaryotes, and the exit site or the E site is also present. The other requirements include the mRNA, tRNA, rRNA and the translational factors. So, what are the roles of RNA in translation? Protein translation by ribosomes requires three types of RNA. The first is the mRNA. The information required for the synthesis of a given protein is present in mRNA. The DNA passes its genetic information in a form of codons to the mRNA to translate it into a protein sequence. The only one reading frame on the mRNA is used for the translation and is selected based on the frame in which the AUG start codon appears. The second is the tRNA or the transfer RNA, and they carry the amino acid and hand them over to the growing peptide chain. The amino acid is bound to 3' end of the tRNA. Each tRNA has a 3 nucleotide base sequence, the anticodon, which is responsible to recognize the codon, that is the complementary basis of mRNA for protein synthesis. And the third is the ribosomal RNA, which makes up the bulk of the mass of the ribosome. One rRNA species, that is 28S rRNA, is a ribozyme that catalyzes the reaction in which the peptide bond is formed. Now, there is two-step process for mRNA decoding. The step number 1, in which the amino acids are attached in the ester linkage to the 3' terminus of the tRNA, forming amino acyl tRNAs. The enzymes that carry out this ATP driven reaction are known as amino acyl tRNA synthetases. So, if we look into the picture, this box shape is the enzyme, that is the amino acyl tRNA synthetases. This is amino acyl tRNA synthetases, and this is the amino acid and this is the tRNA. Now, amino acids are attached in the ester linkage to the 3' terminus of the tRNA, forming the amino acyl tRNA. And then in the second step, the amino acid is added to the growing protein chain based on the codon and anticodon interaction between the mRNA and the tRNA. Thus, a given amino acid often can be carried by more than one species of tRNA. Each amino acyl tRNA synthetase recognizes one amino acid and all of its conjugate tRNAs. Now, the overview of eukaryotic translation. The translation is mechanistically divided into initiation, elongation and termination stages. In initiation, there is the activation of amino acid and the recognition of the initiation codon, that is AUG. In elongation, there is the binding of amino acyl tRNA to the A site. Then there is a peptide bond formation, and after peptide bond formation there is the translocation. And the third step is the termination, in which after several cycles of the activation, initiation and elongation, the stop codons like UAA, UAG and UGA will give the information signal to stop the growing polypeptide chain. And all the stages requires the translation factors in addition to the ribosomes, mRNA and the amino acyl tRNAs. So coming to the first, that is the translation initiation in the eukaryotes. The translation initiation in eukaryotes begins with the three components, and these are the 40S ribosomal subunit, to which the EIF1, that is the eukaryotic initiation factor 1, EIF1A and EIF3 initiation factors are bound. So this is the 80S ribosomal unit, which can be separated into the 6S and the 40S ribosome. This is the ribosomal dissociation. And then there is the bounding of EIF3 and 1A to this 40S ribosomal unit, and this will stabilize this 40S ribosome unit. Then the EIF2, this EIF2 with the GTP and the MET tRNA. For MET, this is the methionine group which is attached to the tRNA. So MET tRNA tertiary complex will form. This tertiary complex will consist of the 40S ribosome unit, the EIF3, EIF1A, this MET tRNA and the EIF2 with the GTP. So this is a tertiary complex which is also known as 43S pre-initiation complex. Now a circular mRNA. A circular mRNA formed by the binding of EIF4. This is EIF4 cap binding complex at the 5' end of this is the mRNA. This is the 5' end of the mRNA to the poly A binding protein associated with the 3' end of the mRNA. And these components of the diagram placing with the MET tRNA in the P site of the 40S subunit. These are placed in the P site of the 40S subunit. Now in the next stage of initiation, the mRNA, this mRNA is scanned in the 5' to the 3' direction until the first AUG start codon is brought into the P site. Then the hydrolysis of the GTP by the EIF2 generates a stable 48S initiation complex, in which the initiator tRNA or the MET tRNA is hydrogen bonded to the AUG codon, that is the starting codon. In the final stage, this is the final stage of the initiation. In the final stage of the initiation, a stable 80S initiation complex is created, and this complex contains the complete exit site, the P site or the peptidyl tRNA, and the A site, that is the amino acyl tRNA binding site, with the MET tRNA bound to P site. So this is 80S initiation complex, which is the final stage of the initiation. Now the second step is the elongation. In elongation, the first step is the binding of amino acyl tRNA to the A site. The tRNA along with the amino acid at 3' end and the anticodon at 5' end attaches at the P site of the ribosome. It only attaches to the codon which has anticodon of itself. After that, another tRNA with amino acid will come and attaches to A site. Now there is the peptide bond formation. After the attachment of another tRNA, the amino acid methionine and the new amino acid will form peptides from each other. Now, after peptide bond formation, there is translocation, and the peptide bond formation takes place by the 28S rRNAs, which is formed from the 60S subunit. After the peptide bond formation, there is translocation, and after the ribosomes moves to the next codon, and the bond between the methionine and the first tRNA will break, and the first tRNA will now be shifted to the E site called as exit site and will leave the ribosome. This step is repeated and all the amino acid will form a chain, and this is known as elongation step. Now, the third step is the termination. After several cycles of activation, initiation and elongation, the stop codon, that is UAA, UAG and UGA, will give the information signals to stop the growing polypeptide chain. And after the formation of after the termination of the information signal, we get a polypeptide chain. Now, after the formation of this polypeptide chain, many changes takes place in the proteins, such as the folding of the proteins, proteolysis, splicing of useless portions, covalent changes, conformational changes, etc., to produce the proteins which are biologically functional, and this is known as the post-translational modification. The process of translation is a complex process, and it has becomes a favorite target for the inhibition by antibiotics like streptomycin, tetracycline, chloramphenicol, etc. Majority of the antibiotics interfere with the bacterial protein synthesis and are harmless to the higher organism. So, this is all about the process of translation. If you like this video, please do subscribe my channel and share with your friends. Thank you for your time.